Alrighty. Got some yerba mate here. And we're going to be looking at Nanome, which is a collaboration and kind of molecule chemistry, biology explorer. I'm going to go ahead and create a meeting here. This whole thing is set up as meetings. I'm on a trial, by the way, but let's create a meeting. Can I create a lobby? Create room. Hugo P's room. Let's make it private. Start a single user room here. And there's a bunch of tutorials I'm going to be going through here. Yeah, I tried this first one, Intro to DNA. And let's do the second one here. What makes caffeine energizing? Okay, we see they have kind of the quest avatars going on. They load a little slowly. But here we're going to be looking at some molecules, so we hit play.
Hey Steve, so as you know, I'm a huge fan of coffee and caffeine. I love tea, an occasional energy drink here and there. So why is caffeine so energizing?
Well, this isn't exactly why caffeine is so energizing, but it turns out that caffeine is actually super similar to another compound called adenosine, which is a byproduct of ATP or adenosine triphosphate, which our body uses as a major source of energy.
It's kind of
Got
cool.
it. So it's kind of like the tired molecule because ATP is energy and this is the by.
So you can pause, and I think you can grab these proteins. Hand or select. I think we want select. Hand. Can I get that? Can I grab it? Room. Draw, torsion, select. Please select a structure. Favorites, recents. Quick save, camera, web browser. Turn my microphone off. Grid lock menus, shared notepad, web browser, camera, quick save. Yeah, so you can actually open a web browser in here. Let's search up caffeine. Go on Wikipedia. There we go. So that is the molecule right there, right, that they're visualizing. Oh, we accidentally clicked on drink. You can select and move this window here. Let's put it right there. Maybe not rotate it. We actually see they've simplified it. There's a bunch of hydrogens that stick out from the side there that they don't have there. So let's hit play again.
So after it uses ATP, it this is what your cells and stuff spits out in order to tell your body that you should get tired now, right? And so I remember that meme. The mitochondria is the powerhouse of the cell, and so the mitochondria produces ATP for the rest of your cell, and then this is what comes after that, right?
Exactly. And so, what happens is you actually have this adenosine A2A receptor right here, which is a GPCR or G protein coupled receptor that sits on the surface of your cell. So you would have your cell out here, and then this would be just on the surface detecting different chemicals. And what it would normally have bound here is adenosine, but the structure that I pulled up here is actually the caffeine. And so what happens is when adenosine tries to go in, but the caffeine's already there, it can't get in. And so this adenosine, when you use a lot of energy, your body's chopping up all this adenosine triphosphate. You end up with this adenosine, and your body will then feel tired when this goes in and binds with your adenosine A2A receptor. But because the caffeine is there, it actually can't get in, and so it doesn't send that signal to your brain to tell you to feel tired.
So backing up a little, just so for those of you who aren't familiar with receptors and cells and stuff like that, your body is made out of organs like the heart and the brain, and most famously your skin being the biggest organ that you have. And so your organs are made out of tissues, which are essentially smaller parts of the organ. And then that tissue is made out of thousands, millions of different cells, right? You've probably seen one under a microscope, maybe like a plant cell in high school or something like that.
Wish I could grab this little thing,
Those
you know?
cells are made out of, millions and billions of proteins, one of which we have here, and a receptor is a type of protein. And so this protein basically sits on the outer edges of the cell, and it kind of acts like a watchtower for this one particular molecule called adenosine. And again, because the watchtower's job basically is to let the body know that it's tired when this thing, the adenosine binds. But because caffeine's so similar, it can bind there instead of adenosine. And it, because it's not exactly the same as adenosine, it doesn't make the body tired, but because it's similar enough, it blocks the adenosine from actually entering, which is just so fascinating.
Yeah, exactly. And so, like Kata was saying, you have this huge cell out here that would just really have this tiny protein on the surface, which of course this protein itself is also made out of atoms and molecules. It's just in this ribbon representation, so we can better see the structure here. It is trying to detect adenosine. This specific protein is called the adenosine A2A receptor because it's out there trying to detect the adenosine. And what happens is it also detects other compounds, that also are similar to adenosine, also bind to it. And so you can see here that this caffeine has made its way in. And there's going to be very subtle changes and modulations between the naturally bound adenosine compound in the A2A receptor versus the caffeine that's bound here.
So this one is the one with adenosine bounded, so that's like the natural state. And then this one's the one where caffeine's bounded. So, we use this metaphor in biochemistry and molecular biology called the lock and key mechanism, where basically these proteins are kind of like locks, and these molecules are basically like keys. And so you can really see when you overlay them like this in VR and AR that, like, basically, just how similar these keys are enough such that then it works on the same lock, right?
Exactly. And so you can see that the adenosine and the caffeine actually are bound in a very similar location here. And so what happens, if one's already bound, the other one, it's not going to have room to get into there because you're already bound in the same spot. And what happens, when these things bind, they all really interact with the protein. And the protein, these things are always dynamically moving, but they're going to end up changing shape slightly, and your body actually uses this as a way of understanding information about what chemicals are out there. So that if the adenosine doesn't bind, then you don't get that tired signal, and that what happened when the caffeine's in there. But it's actually not the only place that the caffeine binds to. So there's actually some other areas that are studied with other proteins, such as this MAO protein, which is
Huh.
monoamine oxidase. And
So
so this is
caffeine binds to an MAO protein?
being a receptor to detect things like GPCRs. It's actually in the enzyme family, where this protein's job is to chop up other molecules and break them down. But you can see that right here we have this very similar motif we talked about has the 5-member ring and 6-member ring with that molecule as well.
The hexagon and the pentagon there, yep.
Yeah, exactly. And so, one other studied mechanism of caffeine is actually binding with these MAOs and actually inhibiting the breakdown of molecules like adrenaline. And so this could be another way that people feel more alert and energized. It's not just that they're feeling tired, but they might actually be building up more adrenaline because they're not able to break it down as much because the caffeine's also interacting with some of these enzymes.
Got it. So this lock or protein, essentially this job is to basically break down adrenaline. But similarly, because caffeine can bound to this lock, caffeine key, if you will, that
It's kind of cool. I didn't know caffeine was a MAOI inhibitor.
Adrenaline. So you literally have more adrenaline flowing around your body when you drink caffeine.
Yeah, exactly. And so it's a small molecule. It's going to spend some time in there, some time out. It's very fluid. You can see larger molecules like this might actually sort of stay in there for longer, or if it's the right molecule, actually get broken down by this enzyme. But caffeine, because it's so tiny and shares that very familiar hexagon pentagon motif, can interact with the adenosine A2A receptor, as we saw. It could bind in this one for a little bit and inhibit adrenaline from getting into that binding site. So yeah, really really cool molecule. I know that you enjoy your caffeine and coffee and green tea and drinks like that. But yeah, I hope that this was educational and kind of diving in.
So we want to see if we can grab this little protein here. I feel like I did it before, but I don't remember exactly what I did. Select chains, select residues, select range, select molecules, select chains. Interesting, we can also mess with this guy's menu. Alright, whatever. We're gonna exit this and then... Oh, there we go. Okay. So we can bring this molecule to us. Can make it bigger. Okay. So this is apparently MAOI, and then we have, can we select molecules? Yep, that's what we want. We want this one. Molecule 267. Just make it big so we can actually sub-select here. I think this whole thing might be glued together. So if we draw... Damn, I didn't know you could draw. Oh. So this is kind of the little caffeine, apparently. I think the caffeine molecule itself was a separate molecule. this is all just one piece, so we can't separate it out. But it's good to know. Now we can go back. Shared notepad. Let's close out of the web browser. Presenter request. Quick save. Exit. Return to lobby. Don't save. Okay, so we're gonna go do the second tutorial, see if we can actually make it single user. Let's see, maybe if in some of these next tutorials we can actually Chemistry 101, amazing flow of information, 7 minutes. From DNA to RNA. Alright, let's try it out. We can move this menu over here. Okay, this guy's talking. We can... How come we can't hear him?
Get the central dogma. Overall, the central dogma is the explanation.
No, he's not muted.
Of cell. In the first macro.
On mute? Okay. Looks like this guy is muted. Okay, he has a molecule here that he wants to grab. See if we pause. If we exit that, we can probably grab his molecule. Here we go. What was he looking at? He was looking at DNA. So this is DNA here. Right, you have this kind of backbone structure here. And then you have the different base pairs here. So yeah, you have one base pair here. I wonder if I can select residues, atoms, residues. All right, so DNA G, DNA H, DNA G. So H and G always go together, right? This is also H, and this is also G. It's also H. Let's see if we can find an A. H, H, H. We're looking at this wrong. DC, DC. Yeah, no, it's not. ATGC are the three types. Maybe we want chains. Yeah, that's what we want. So H, G, H, G, H, G. Let's see if we can find an A. H, G, H, G. These are only the same? Maybe it's not chains what I want. Maybe what I want is select atoms, range, molecules. Okay, this whole molecule is getting selected. Okay, the whole thing gets selected again. Yeah, it's basically the two sides, so you got one and then the other. Can we select residues? When we select residues, then it's just this part here. Right, it includes the pair, I don't know what they're called, I think base, and then it also includes part of this backbone here. So you see how the highlighted ones are the actual base pair there, and then the backbone. So maybe when it says DA, it means A goes with T, DT, A goes with D, select one here, G goes with C. Okay, we finally found it. So it's the second letter in this. When you go to select residue, it'll select this whole pair, but it'll also select the backbone here, or this little part of the backbone. Actually, we can figure out what that is. Atoms. What is this atom? Phosphorus, oxygen, 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 carbon, 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 oxygen, nitrogen, carbon. So these blue ones are nitrogen, these gray ones are carbon, and then these red ones are oxygen, and then these big yellow ones are potassium, I guess. So, right, we were saying this here is A and T, right? So the A has this kind of two, like one 5 ring, one 6 ring. The T just has a 6 ring. And this also has a little nitrogen sticking out. The T has a little oxygen, 2 oxygens and a carbon. And then if we go up to the, which one? This one here? C, A. And now we're still in atom selection residues. That's what we want. The T and the A, the C, and the G. Okay, so this pair here are C and G. So similar but different. This one, if you actually look, this one, C and G are much more similar. Or they're not. You still have the same kind of situation where you have a 5 and a 6, and then this one has a 6, but this one has the, let's select atoms, oxygen, nitrogen, oxygen, nitrogen. So this oxygen and this nitrogen attract each other here. Okay. Let's exit, and then we can go back to the actual create room. Private, start single user room. Display menu. Modify. Nanome tool select, medchem, torsion tool, docking, mutation, and advanced selection. Let's go over the tools. Why can't we hear anything? You, please, talk. Okay. Entry list. Quick menu, load, color, display, entry. Oh, he's got control of it. Okay, so you can actually measure with this. Distance, delete, move, angle, dihedral, distance, delete, move. Okay. Spatial tutorials.
Welcome, little blue dot right there. This just allows you to twist around rotatable bonds. The medchem tool is awesome because you could
Maybe this is
actually
outputting the volume
create
to
new
something.
molecules.
No, I mean it does look like it's there. Okay. Seems like you can draw. That seems kind of cool. Why don't we try to draw something? Okay, if we exit out of this, take this guy's menu here. Take this molecule. Bring this closer to us. Take this molecule. Can we measure? Can we measure? Delete. Go to hand. Select molecules. Oh, you can turn it. Cool. Okay, spatial tutorials. Have that open. What about this one, entry list? Close out of that. Molecule. Can we trash this? What is this? Delete your current selection. Yeah, we want to delete. Okay, so we've deleted this molecule. Let's go to the modify menu. We'll open it up here. Let's draw. Let's bring this over here. Oh. Okay. So I wonder if we go to a web browser. Maybe we can go to our history and history. Caffeine on Wikipedia. This one. Okay. Right. I wonder if we can. Okay, cancel. Draw. Let's just try to do this. Is there a way to like export this? Or this is literally just drawing? Just drawing. Alright, we're just gonna get rid of that. This is worthless to us if all it is drawing. Macro, modify, advanced, mutate. Out of that. Minimize, clash. Export, save. Please select a structure. Stacks. Plugins, okay. Modify. A tool that allows small molecule building. Build. Okay, we need a this one here, we need 3 of these. Trifluoromethane. Actually, maybe not. Let's just try to build this from scratch here. So we have 1 carbon. How do we place it? Let's go back to the build menu. Modify this menu, put it over here. Then we will take the build menu, put it here. We're going to medchem. Here we go. You can look at the entire periodic table of elements. We just want the atom C. How do I place this? Property view, small, big. Okay. Residue view, medium. What if I just... Why can't I, like, place it? Hold to create new molecule. Okay. Let's go back. Okay. We put the little carbon atom. Close out of the web browser. It seems like it's dying. Web browser. History. Caffeine. Okay. Come on now. Clear all. Yes. Yes. Enter. Caffeine. Enter. Okay, web browser is not working anymore, so what we're going to do here is we're actually going to exit out and then re-come back in. Delete everything. And then we're going to go room, return to lobby. And we are going to create a room, private room, start single user room. Okay. We're in an empty scene. Let's move this. Let's try the web browser, make sure that's working. See if we go to history. See if we do. Caffeine, enter. Get the Wikipedia. Okay, now we're going to try to build this. So let's go to the modify menu, edit. Nope, we want the, let's close out of this entry list menu. Close out of that. We're going to go into quick menu. Modify. Okay, we're gonna. Oh shit, they have some. Design a molecule different. That's cool. First, let's try to make caffeine though, and then we'll try to do something cooler. So let's do modify, tools, medchem. Then we're gonna do build. Close out of this crap. We're gonna start with a atom, a 1 carbon atom. We're gonna place it. Okay, make it a little bit bigger. So then we need a double bonded oxygen. So we're gonna put double bond with oxygen. Okay. Then we're going to have a nitrogen. We're going to have one here. And we're going to have another one here. Let's go back to carbons. We're going to have one carbon here. One carbon here. And one carbon here. Okay, then we're gonna have one carbon here. And then we're gonna have 2 nitrogens here. And then it looks like a carbon at the end of each of those nitrogens. So there's gonna be one here. There's going to be one here. Or no, this one doesn't exist. This one should be here. And then there's going to be another one here. Okay, so now let's create these double bonds. We have a single bond here. We have single bond there. Undo. Okay, it looks like these are a little bit too... Can I like select? What if I make this smaller? Then can I modify tools, medchem, single bond? There should be a single bond between these two. There we go. And then there should be a single bond between those two. Okay, so I think this is actually more how you build them. And then you do a single bond to another carbon. Okay, it autofills the carbon in there for you. Then there's another single bond to a nitrogen. This carbon has a double bond to an oxygen. Alright, this nitrogen has a single bond to another carbon. Okay, so we're doing well. We have part of this already built here. This nitrogen here has another single bond to a carbon. Good. Then we have a single bond to another carbon. Then we have a double bond between these two carbons there. It auto determine the shape for you. That's pretty good. Then we have a single bond and another single bond here, except both of these are nitrogens. Then we're going to have a single bond to a carbon and then a double bond from the bottom nitrogen to this carbon. Oh, nope. Undo. Between that. Alright, there we go. And this top one here is going to be a single bond into a carbon. And these have like, what are those little hydrogen boys? Hydrogen. Let's put a little hydrogen there. Close out of that. And these are a bunch of little single bond. Oh no, I think this is it's not a single, double, or triple bond, it's a affinity or something like that, right? A bunch of crap here. I wonder how we can we delete, group delete, and select, or no, we can actually probably come here. So if we select the chain, okay, so how do we unselect? So you click it again to unselect. So let's select all of these. Let's delete them. Okay, so we have our molecule and then we have our second molecule. Let's bring this menu closer. Sequence align. Select a chain. Okay, I guess that's already aligned. Our little caffeine molecule here. How do we get this select molecules? How do we get that little carbon to interact with this ligands? Okay. Can I save this? Molecules. Advanced Metadata. Okay, so this is to create animations, but we don't really necessarily need that. Okay, this is cool. Let's open a cool workspace here. No, we don't want that. We want entry. Oh, wait up. Not stacks, display. Okay, so we have atom, ball and stick. We can just go pure stick. We can say van der Waals. This is a kind of a visualization. Display the selection in van der Waals representation. We got wire. Adapt to color. Ball and stick. Stick. I kind of like the ball and stick. Substructure. Hydrogens. You need a commercial license for the hydrogen, so those are these little ones here. That's how they would be on there. Okay. Let's get rid of that. Alright, here is the load. This is what we were looking at before. Avatar customization, we don't really give a shit about that. Molecular dynamic simulations, electrostatic potential maps, kinase structure, COVID-19 main protease. This is the dual leucine zipper and kinase as an inhibitor. Kinase. Let's load that. Holy shit, this is cool. This is the AMP kinase, so let's go into our selector here. Molecules. Let's make this big. Let's select individual residues. Nope. Select chains. Nope. Can't do anything. Oh, maybe we can. No, select atoms, residues, chains, range. No, we can't seemingly select any of these things, but let's take this load menu and this molecule's getting in our way here. Let's put this there, put the load menu's locked. Let's put the load menu right here. Let's take this, bring it a little closer. Put it right there. Lock it. Let's search kinase. Catalyzes transfer of phosphate groups from high energy phosphate ATP into ADP. Okay, so ATP triphosphate becomes, yeah, so ATP becomes ADP, and this is basically, as far as I understand, this is like literally all energy of life comes from this. So let's zoom in a little closer here so we can see better, right? So you have ATP, which I guess is one of these. Maybe not. Doesn't quite look like it. And it becomes ADP. It loses one of those phosphate groups. Doesn't look like it. I think it might be a different kinase. Let's close out of that. Dual leucine zipper. Sequence menu, macro scripts. What about electron density map? Let's move this over here. Let's bring this over here closer. Let's unlock it. Bring it closer. You can load molecules with an electron density map from RCSB. Okay, so there's some way of loading these formats. Okay, there's a CIF format. That's the format we were looking at this morning. It's a crystallography format. You can grab the EDM display area the same way you mold a molecule. Okay, point your laser bolt and hold down the grip. Okay. Okay, so this is basically the electron density. So electrons themselves, right? A lot of people think of them as particles, but they're also a wave, right? So they're not in one specific place at any given time. There is a distribution, right? That you could think of it like think in your head like a little Gaussian curve, right? And the electron is anywhere inside that little Gaussian curve where it's more likely to be in the middle than on the ends, right? And now take that Gaussian curve and make it 3D in your head. And this is kind of what you're looking at. You're looking at areas where the probability of there being electrons is high and areas where the probability of there being electrons is low. So obviously when you have these type of molecules here that are kind of bonded to each other and you have all these interactions between other molecules, there's areas that are going to have high electron density and other areas that are going to have low electron density. Okay, so flat map, smooth. Okay. So we can take this menu here. Let's bring it closer to us here. Okay, I wonder what this means. There's two different here. There's red, green, and blue. What does these mean? Find the entity containing EDM that you have loaded on the entry menu. Click on the second button from the right on the same row. This is the advanced. Okay. Okay. That's kind of all they're saying. Cool. Let's close out of that. We lost our little entry menu. Where'd it go? Ah, there it is. Entry, load, featured. Source. Oh, they have the protein database. Stacks. Macro. Entry. Stacks. Fuck, I keep forgetting how to get to the workspaces content. That's how you get to it. No, that's not how you get to it. the workspace. Structures, pictures, featured. Featured workspace, load featured workspace. That's what we want. A protein sequence, export molecules, design a molecule, share a picture, macro scripts. What about simulations? I think they keyframe animated these. That's kind of cool. So this is kind of a molecule, kind of showing you all the give. So, right, with any of these structures in reality, they're moving. So you can actually see, for example, here, look at this, look at this little, how it rotates, right? Let's unlock that, bring this forward. Playback. (100x speed). Let's do 1x speed. Clash, mutate, advanced. Minimize. Measure distance, measure angle, measure distance. What do we do from here to here? 5.79 atomic units. And then if we measure from here to here, it's 16.03 atomic units. Quite cool. Take a pic. Sharing. Testing molecules. Send. We'll see if we get it via email. Cool. I think that's a little exploration of the Nanome app. Obviously, I don't know anything about biology, to be honest, so I'm kind of just trying random things, but pretty cool little app, you know? We'll see what happens, and we'll see if we can make use of it. All right, thanks for watching, and see you guys later.
Yeah.